Okay, so this is going to be the final endocrine system video. It should be relatively short. Is good. I'm just going to cover a couple of the neater, more wacky, I don't know, different sorts of hormonal events in the lives of animals. Okay, so it won't take too long, but will require us to move forward with that. So, okay. So one of the first kinds of interactions, and this is going to lead us into the next chapter here, that we should talk about are both sort of sexual interactions, but also pair bonding interactions. And here we go back to the hormones oxytocin and vasopressin, right? So posterior pituitary hormones. Both are released in mammals anyway. Both are released in response to the act of mating. And they have different effects. Well, they have the same effect, but different hormones do it in different organisms. In mammals that form pair bonds. Again, so not every mammal forms a pair bond, but in mammals that do form pair bonds, the pair bonding behavior is reinforced by oxytocin in the female and by vasopressin in the male. They both work essentially in the same way. They both cause sort of the stimulation of reward circuits. So the same thing that makes drugs addicting and things like that. But in response to, is obviously being around the other individual, forms these sort of pair bonding links from that. So that's what love ultimately is vasopressin or oxytocin, depending on your sex, stimulating those reward circuits for you. Other relatively interesting mating type behaviors in amphibians, you may or may not be aware, I suppose. They don't have the same kind of moving parts that we do. They have a cloaca, both male and female, and so the only way for them to fertilize the eggs is to simply bring their cloacae nearby each other and kind of release them into a cloud in the same general area and hope for the best. So in order to kind of facilitate this, a lot of amphibians do this sort of behavior. It's called amplexus, A-M-P-L-E-X-U-S, amplexus. And the male literally climbs on top of the female and kind of puts her in this sort of wrestling hold. And this can last for hours, right? It can be a really prolonged kind of experience. And that's fine in and of itself, but of course, remember that they're vulnerable to predation at this point, right? Obviously, the female's not getting away anytime soon. The male's kind of locked in place too. There's two of them, which makes a more enticing meal than just one. And so there needs to be a circuit or a means by which they can rapidly detach from this amplexus and go their separate ways. And this is where we come back to things like corticosteroids. Okay, now, corticosteroids actually release the amplexus not directly, but they act through neurotransmitters. They act through endocannabinoids, which is a class of neurotransmitter. From the name, you might have guessed they are in the same family of chemicals as tetrahydrocannabinol, THC. But what these cannabinoids do here is they actually inhibit the neuron that stimulates the clasping. And so by inhibiting that neuron, clasping is broken and then shoot off in opposite directions fairly quickly. As far as chemical messengers themselves go, not specific here now to hormones or even neurotransmitters, we find that they operate on kind of a continuum of distances, right? So from things that are relatively short, okay, almost essentially non-existent distances, things like gap junctions or CAMs, sort of cell adhesion molecules, things that stick cells together, to relatively short distances, things like neurotransmitters, okay, and then from there to paracrines and autocrines, right, relatively local, okay, not no distance, but local, okay, to things like hormones, which can travel the entire length of the body, okay, but also to things like pheromones and chiromones. We talked about pheromones before when we talked about various sort of accessory chemical senses, things like that, as being messengers intentionally put out by an animal for other members of its same species, or potentially for others, as a communication kind of a thing. A chiromone is a similar idea, but it's not for communication. It's simply just a chemical cue given off. And in fact, sometimes these can be quite bad. This is how parasites find hosts in a number of instances, for example, is by following chiromone cues. And so with chiromones, we can talk about allomones, okay, which are these chemical cues released for self-benefit or benefit within the species. And then cynomones, which are like what the parasite would use to find a host, right? Not for the benefit of the species releasing them. Sometimes for mutual benefit, sometimes not. Outside the vertebrate realm, the hormonal system we understand best is the system of ecdysis. Okay, this guy right here. Ecdysis is just process of metamorphosis and shedding. Right? So remember, these are invertebrates. Their skeleton is entirely external, and so in order for them to grow or to change shape, they have to shed that exoskeleton so they can expand beyond its limits. And this metamorphosis, this development, can be dramatic, right? Like we see what we'll call a hollow metabolist development, where we undergo a massive metamorphosis from a caterpillar to a butterfly, or it can be hemimetabolous, where it's not 100% isometric growth, but there's no dramatic metamorphic event, right? The nymph, various instars, these juvenile stages, I call them instars and invertebrates, are not terribly different from the adult. They're different, but they're not tremendously different. So here, for example, we're seeing the development of wings in the adult, but not in a lot of these earlier, earlier instars. For the dramatic ones, for the holometabolous ones, there's usually a lot of different cues that go into how all these stages play out, right? So for example, with our silkworms here, the eggs themselves are responsive to a cue, right? And this particular cue is actually external, it's temperature. So they're not going to hatch until there's been so many days above a certain temperature, okay? And the reason for that is that the early instars, right, these juvenile stages of these silkworms, only eat mulberry leaves. And in order for there to be mulberry leaves for them to eat, it has to be a certain days above freezing in a row or what have you. So that's the first cue that we're getting there. Now they're going to eat a bunch of these mulberry leaves, right? Eventually they're going to spin a cocoon. Okay, this is where the silkworm comes from. And then inside the cocoon, this is where this massive metamorphic behavior is going to occur, right? And the reason they're going to change from a juvenile to an adult stage is that the adult stage is specialized primarily just for mating, okay? Really for any other purpose, they're specialized here just to find a mate, to lay eggs, and to start the cycle all over again. The hormones that are responsible for these different stages, there's a number of them, but there's really two that are super important for us. We have juvenile hormone, which, as you might guess, is a hormone that kind of maintains juvenile characteristics, right? And you can see here it is not a steroid hormone, right? And then the one that's going to stimulate the ecdysis itself, right? So 20-hydroxyecdysone, or simply just ecdysone for our purposes. Ecdysone, the steroid hormone, this is going to be the responsible thing for all of this, right? All of the behaviors associated with pupating, with metamorphosis, with emergence, driven here predominantly, not exclusively, but predominantly by ecdysone. There are a couple of other relatively important hormones here for us. ETH PETH, PTTH. I'm not going to go into what all of those stand for, prothoracotropic hormone, PTTH. I don't care that you worry about that. What they're going to do, though, their relationships, though, are built around a lot of the things that go into ecdysis, right? So in the brain, for lack of a better word, of our insect, we have these two long extensions, and these are actual things. Like here's an actual micrograph of these structures here. But these structures, the distal ends of them, are responsible for the production of two different sorts of hormones. Okay, so the epithelial component of it produces juvenile hormone. All right, so this is the thing that's keeping it at a instar stage, at a juvenile anatomy. The neuroendocrine part of this, though, okay, is responsible for producing PTTH, this prothoracotropic hormone. And this, right, it's a tropic hormone, right, stimulates the, as you might have guessed from the name, the prothoracic gland, okay, which in turn produces ecdysone, right, this thing that's responsible for the ecdysis itself. Now, the other tricky part is that once we have begun ecdysis, once we're started to try to get rid of this old exoskeleton, we have to get it off. Right? We have to get it out or get out of the old exoskeleton somehow. And that's where ETH and PETH come in. These hormones stimulate the stereotypical behaviors, the sort of the wiggling out of behaviors that we associate with the actual shedding of that old exoskeleton. And they are linked here. We haven't talked about this yet, but this is the respiratory structure of the insect. And so when they are essentially blocked over, this is a stimulating cue for the release of ETH and PETH to begin trying to shuffle off that old exoskeleton. Okay, so that's this for that. That's that for this chapter. The next set of videos we'll be talking about sexual reproduction, and then from there, who knows how long we're going to be doing this, but we'll at least get through those two chapters here. So join me for the next unit, I guess.